Hello, virtual geography cohort. As you can see from the little box with my face in it, it's Poppy, here to present my project proposal to you people. The generalized title for my project is Benthic Communities of the Charlie Gibbs Fracture Zone. I'm a master's student here in the Department of Geography at Memorial University of Newfoundland, and I'm going to take you on a virtual journey to the deep sea. So I'm going to start with an introduction to my study site, which is the Charlie Gibbs Fracture Zone, hence the title. And this fracture zone is a transform fold, which is like a big crack in between a tectonic plate boundary. This particular one offsets the mid-Atlantic ridge in the North Atlantic Ocean. As can be seen from this image here, it's pretty big and separates the warm waters of the Azores with to the south and the cooler waters surrounding Iceland. This creates what can be described as a biogeographic boundary. These can be any physical boundaries created by a geographic feature in nature. In this case, a continuous underwater mountain range, which can create a separation which can create separations in species in some cases. The Charlie Gibbs Fracture Zone itself has been recognised as a very important geological and potentially an ecologically important feature of the North Atlantic Ocean. This is reflected in the efforts that have been made to protect it from harmful anthropogenic activities by establishing a marine protected area, which can protect marine habitats from fishing, mining, and basically anything that's damaging to the natural habitats of the area. So here we have an image that shows you the boundaries of this marine protected area. There's a lot going on in this image, I know, but if you just focus on the yellow box that's labeled Charlie Gibbs Fracture Zone, the shaded part is under full protection by law, which includes the seafloor and the water column. So anything from the surface of the water all the way down to the seabed, including the seabed and what's in it. The non-shaded part of the Charlie Gibbs Fracture Zone square is not fully protected and is only the water column is protected. So from the surface, sea surface to just above the seabed, and the benthic communities, the benthic habitats that reside there are not protected at all. So they can be exploited or basically they can be damaged by human activities. We know very little about the different biodiversity in different areas of this feature. There have been past studies. One in particular was called the EcoMar project, which looked at benthic communities in the north and south of the Charlie Gibbs Fracture Zone. This highlighted the difference between the communities either side of the fracture zone, so on the north and the south, but gave little insight into the communities which inhabit the fracture zone itself. There have been transatlantic surveys which mapped bathymetry maps of the seabed itself, but didn't look at the biology. So basically, there's very little information on the biology of the Charlie Gibbs Fracture Zone. This last image is where the survey where I got my data. That's where they did their bathymetric mapping. So the problem. After diving into previous studies as well as legislative history surrounding this remote area, I have identified a gap in the knowledge on the Charlie Gibbs Fracture Zone. The structure and composition of the benthic communities is virtually unknown for areas on the fracture itself. Since my study site is located just outside the boundary of the fully protected southern part of the protected area, the description of the biodiverse benthic communities could be an integral part in the future of the decisions made on this for its protection status. So yeah, the gap in the knowledge is a huge thing. We don't know anything about this area that I'm looking at, and its protection status. So the communities that I will be classifying, identifying and classifying are currently unprotected. So that is a problem that I'm going to be addressing, hopefully, as well, that my project will address. So the purpose of my project is to address these problems using a more I'm going to redo this one. To do this, I will conduct a detailed Benthic community analysis in order to help fill this gap in the knowledge for the Trigus fracture zone. To investigate the relationship between the environmental variables, so things like substrate type, which can be like if it's rocky, or if it's like in this picture here, or if it's soft sediment, so like sand or mud. Other environmental variables include like how rough the terrain is, so how like rugged it is, which can be it's measured from the multi-beam data, and it can basically just mean how structurally complex the seabed is, basically. Yeah, so there's some of the variables that I'll be looking at and seeing if they're important for how the community analysis is structured. Yeah, basically in order to help fill the gap in knowledge. To investigate the relationship, oh yeah, I'll identify the habitat forming corals as well and assess the effect that they have on the biodiversity of these habitats. So that'll be looking further into how biodiversity these habitats are and what drives that specific level of biodiversity. Excuse me. I will then classify the habitats under the OSPAR guidelines. So OSPAR is it's a non-governmental body, but it basically creates recommendations for how to manage the North Atlantic Ocean. So it deals with all of the spatial planning, the marine spatial planning. So that is like marine protected areas, essentially. Yeah, it deals with protecting the most important areas of the North Atlantic Ocean, and it is mainly deals with the European countries that border the North Atlantic Ocean and Iceland. Yeah. So I'll classify the habitats based on their guidelines, so that it can be used in the future decision making for the North Atlantic spatial planning. So for my methods, my data was collected aboard the TOSCA survey, TOSCA being short for Tectonic Ocean Spreading at the Charlie Gibbs Fracture Zone. This survey was mainly focused on assessing the geological composition of the region, so it was a geology-based survey. The remotely operated vehicle, or ROV, Holland 1, was deployed to collect video footage on 5 transects. So there was 1 transect per ROV dive, as well as geological and biological samples were collected from the deep sea floor as well. The shipborne multi-beam echo sounder created detailed bathymetric maps of the seafloor, which provided a visual representation of part of the fracture zone. This survey was conducted aboard Ireland's research vessel, the Celtic Explorer. So here's a picture of the ROV being deployed off the Celtic Explorer there. My part starts with the video annotation. So first I built a species catalogue, I built this catalogue from the images that were taken while the ROV was recording video. So I created with these images every single species that was unique. I created this species catalogue. It's basically just a hierarchy of folders which represent the species that I will encounter in the video, and what groups they belong to. So this is an example of what it looks like. This is like all the species of sea star that I saw. So that's an example of what my species catalogue looks like. So I'll record every organism that I see as an observation in the video annotation software, which is called VARS. My species catalogue will be copied into the knowledge base in VARS, which looks like this. So it'll translate into this knowledge base. This is what I will use directly to annotate my video. For my annotations, I will derive a species matrix using the R program. Sounds very fancy, but it's really just a huge Excel file with all the species documented in it every time they were observed. So it's a very big file and looks very scary, but once I run it through statistical tests and I'm able to manipulate it in R, then it becomes a lot easier to manage. My statistical analysis will begin by using an ordination technique which will help me to order my observations. They'll order the observations as points on a 2D coordinate frame. This will allow me to visualize potential groupings of organisms which share similarities. For example, they are found on a similar substrate type. I will perform a variety of multivariate tests to determine the true community assemblages. I will derive the terrain variables, which are things like rugosity and things that I explained earlier, using the multibeam data in ArcGIS. For my classification scheme, I'm going to use the UNIS classification scheme for the substrate types, which is a standardized system used throughout Europe, and it's used in any decisions regarding protected areas. So it's a standardized one for this kind of study. For my habitat classifications, which I will use my community assemblages that I created for my statistical analysis. I'm going to use the OSPAR guidelines for the classification as my study site is within the OSPAR governed region. So my expected results. I expect to produce a detailed description of the community assemblages that I've observed from the site on the Charlie Gibbs fracture zone, which will shed light on the diversity within this feature. I hope to determine the environmental drivers behind these community assemblages and identify the important habitat forming species of coral, which may also contribute to the community structure. I expect to quantify the biodiversity of these sites, as generally the more biodiverse a site is, the more likely it will be protected under something like the OSPAR Commission. I expect to create a list of classified habitats under the OSPAR guidelines that will be easily interpreted by decision makers into the future. I expect these outcomes to be useful in any future decision making on the protection of benthic habitats of the Charlie Gibbs fracture zone from activities such as fishing and mining. So this project will add to the knowledge of the benthic habitats in the Northeast Atlantic in general, as well as providing the first description of benthic habitats and their drivers on the Charlie Gibbs fracture zone. These descriptions will potentially play a key role in the future protection of this important feature and the habitats which populate it. Protecting these vulnerable habitats is vital if we are going to maintain the health of our marine environment. I'd like to thank the Tosca team and the crew of the Celtic Explorer for all their hard work collecting these data. And that's it. Thank you for listening.